Assalamualaikum. My name is Aqil bin Muhammad Anwar. My matric number is 23DBT20F1017. Okay, today I'm going to present about simulation SDS page. Next, theory or principle of SDS page. So what is SDS? So SDS is stand for sodium dodecyl sulfate polyacrylamide gel electrophoresis. So the theory behind this SDS page is to separate protein based on size through the use of a stacking gel and resolving gel. And the principle of SDS page states that a charged molecule migrates to the electrode with the opposite sign when placed in an electric field. So the separation of the charged molecules depends upon the relative mobility of charged species. The smaller molecules migrate faster due to less resistance during electrophoresis. Okay, so for the method, materials and why we use the materials. Okay, firstly, we will use SDS polyacrylamide gel electrophoresis, which is SDS page, because of the separate proteins of cellular extracts, which contain all proteins of a cell. Next, the extracts we are interested in come from two CRISPR-edited macrophage cell lines. We also need to include appropriate control for this experiment. Controls will help increase the reliability and reproducibility of results. So the control, first, positive control. Positive control is a sample in an experiment that produces a known result to compare with the test sample after the same treatment. It is used to control for unknown variables and confirms that all your regions work. And the next control is negative control. The negative control is the opposite of the positive control. The positive control produces a non-result, whereas the negative control is designed to produce no response after the same treatment. That's all for my simulation of SDS page. Thank you. And after this, I will show the simulation. Okay, now we will start with the first step, which is collect cells. So the first step for collect cell is increase the heat block temperature to 95 Celsius. Next, we will set a micropipette to 30 UL and attach a tip. Okay, after that, we will draw up the white type cell culture. First, we attach a tip first, and then we'll draw up the white type cell culture. Next, close the wild type cell culture. After close the wild type cell culture, dispense the wild type cell culture into the wild type cells tube. Open the lid and put the wild type cell culture in the tube. Next, we will repeat the procedure to transfer 30 UL of the pancreas cells culture. Before that, we will attach a new tape before we continue to the pancreas cell culture. after draw the cell culture, I will put that in cell culture tube, pancreas tube. Next, we will repeat the procedure to transfer 30 UL of the edit for cell culture into the cell 4 tube. Finally, we will repeat the procedure to transfer 30 UL of the edit 7 cell culture into the 7 cell tube. We must remember that we need to attach a new tip before we draw up the new cell. Alright, done for the first step of collect cells. Okay, the second protocol is add sample buffer. So the first one is we already set a micropipette to 10 UL and attach a tip. Next, we will draw up 10 UL of 4 times SDS sample buffer. After draw up the 4 times SDS sample, we need to close the 4 times SDS sample buffer. Next, we will dispense the SDS sample buffer into the wild type cells tube, and we will repeat the procedure to dispense 10 UL of 4 times SDS sample buffer into the pancreas cell tubes. Always remember that we need to attach a new tip and inject tape in the biohazardous waste. After done the procedure, we will repeat the procedure to dispense 10 UL of 4 times SDS sample buffer into the edit 4 tube. And next, we will do the same thing at the edit 7 tube. Done for the second protocol, and the third protocol is demature the protein. So the first one is we need to move all our 4 sample tubes to the heat block, and next we will set the heat block timer to 10 minutes and press start. And finally, we will remove all our sample tubes from the heat block to the tube rack, and we will move to the fourth protocol, that is centrifuge the samples. We need to move all our sample tubes to the microcentrifuge. We need to put the sample in balance. This one it's not balanced. I will arrange it and balance. When the centrifuge looks balanced, we will start and spin the tube. After done the spin the tube, we will remove the tube. Next, we will go to fifth protocol that load the gel. Okay, first we'll place the polychloride gel in the electrophoresis box. We need to open the lid and put the inside. And second, we fill the gel cassette with 1 time running buffer. We will put the 1 time running buffer in the gel box. Next, we will remove the comb. Put it back and we will remove the comb. Remove comb. After remove the comb, we will fill the outer gel with the electrophoresis tank with 1 running buffer. And we will set a micropipette to 20 UL and attach a tip. After we attach a tip, we will draw up the protein ladder. After the draw up the protein ladder, we will move the micropipette to the first length of the protein gel. The first one. And click next. Only press into the first press. Okay, so the pop-up question, I will answer the slowly to prevent it spilling over into the next one. Alright, submit answer, and we will continue the procedure. Now we will dispense 20 UL of the positive control into the second well of the gel. Attach a new tip, eject the just now tip and attach a new tip. We will draw and dispense positive control into the second well of the gel. First we need to know that the positive control is what type cell. Put it in the second well gel, and after it we should know that negative control is the pancreas tube, and put it on the third wall of the gel. And then we will dispense 20 ml of the added 4 cells into the fourth gel well of the gel. Finally, we will dispense 20 ml of the added 7 cells into the 5th well of the gel. Okay, so there is last procedure for load the gel. Then we will go to the next protocol, that conduct protein electrophoresis. We will cover the electrophoresis box, and we will connect the red and black cables to the power supply and the lid of the gel at the electrophoresis box. Black to black, red to red. And we will turn on the power supply. After we turn on the power supply, we will set the voltage to 150 volt. Next, we will set a timer to 60 minutes, and then we will start the power supply. After we start the power supply, wait until the time has elapsed. So there is pop-up question, and the question, how do you know if the gel has the... So the answer is the answer So the answer is C. The dye front is at the bottom of the gel. After that, we will turn off the power switch and unplug all the electrodes. Put it back at the place. Make sure that our power supply is switched off. And we will inspect the gel. And we need to inspect the gel. And we have done the protocol of the experiment. Thank you.